Okay, thanks. So first, I'm sorry that I cannot give an in-person lecture due to business matters. And so anyway, so since the audience is not that big, so just interrupt me if you uh, have any questions. So thanks the committee for the invitation, and thanks Dr. He for the introduction. And it's my great pleasure to present our recent work about network reconstruction using causality measures. So actually, the talk consists of a series works on this topic, and including some PhD students at SJTU, and also some faculty members from NYU, and also some experimentalists from this ECNU who helped to verify our methods. So actually, Yan Xiao is now a faculty member at the Shenzhen Institute of Advanced Technology, and he focused on the simulated networks of Hodgkin-Huxley and leaky integral phi models. And later, Kai and Zhongqi extended the theoretical results to real data. So okay, so we know that different species have different brain in terms of the weight and also the neural numbers. But the common feature of all the brain is that it is a multi-scale system. From this nanoscale, this synapse, meaning the neurons connection site, and to the micro scale, this single neuron, or to this neural circuits, this millimeter scale. So, and thanks to the development of experimental techniques, now and also along with computational power and artificial intelligence, now we have accumulated a large amount of experimental data and phenomena, which allows us to do very detailed data analysis. However, as nature and one of the special issue released by nature, so there are challenges in irreproducible research in life sciences, especially in neuroscience. And the reasons are quite diverse, including this sample size and experimental condition, and also this software error, and also this statistical analysis, abuse of statistical analysis. And so the solution actually was proposed by one is from this Hinton, and the other is from Sinofsky, which means you have to do very quantitative math, including modeling, analysis, and simulation that can essentially extract the mechanism underlying the complicated experimental phenomena and data analysis. So that's actually the central goal of computational neuroscience, which is my research field. So our lab actually focus on the mesoscale of the brain, meaning we start from single neuron and to form neural networks. And what we are interested in is the relationship among the network structure, dynamics, and function. And ultimately, we want to extract some quantitative principles by investigating these brain networks, and which later can be used to design brain-inspired artificial intelligence. And this includes the intersection between experimental neuroscience and applied math. So, so our lab actually treat the brain as a complex network, and we studied from three aspects. So first is at this network node, meaning single neural network, we have investigated the quantitative rule of how single neuron integrate multiple synaptic inputs and how to infer the inputs, meaning the positive or the negative current received from neuron by its output. This can help us to determine the neuron state. And recently, we also studied how to build a precise and efficient mapping between biological neuron and artificial neuron. And at the second level, meaning network structure level, we are interested is in what is wiring diagram, meaning what's the common feature of this wiring distribution, wiring length distribution of brains of for different species, whether we can extract the common rule for this for the brain of different species. And another thing is we are also interested in how the brain just keep in balance. Here, balance means the neuron receives both positive and current and negative current, and somehow they are statistical balanced, which is very important for our brain to perform function correctly. And once we have a single node, meaning single neuron, we have the neural network structure, and then we can build this large scale model for certain region or even the whole brain region. And what we have investigated the encoding and decoding principles of early visual pathway and early olfactory pathway. And so whether there exists compressed principle in our neural networks which can efficiently transfer the information from upstream layer to the downstream layer. So today's topic focus on the second, meaning the network structure level. What we are interested in is in actually the inverse problem, meaning so how to infer the neuron's connectivity structure from the outputs we measured from these neurons. So before that, let's first look at how in experiment to obtain this connectivity structure. So human connectome project actually is the main benchmark and the data source for the structural connectivity among coarse-grained brain regions. And meanwhile, for the microscopic, this cellular level connectivity, complete this projection diagram of this mouse prefrontal cortex was obtained. And it demonstrated actually for single neuron, it can project multiple areas. And later on, and a complete wiring diagram of the adult fruit fly was obtained. It actually sets a precedent for the later large-scale connectome program, because we know in biology, the function is normally determined by the structure. So in order to understand the function, it is important to know the structure of the system. So here, for the brain, for the neural networks, the structure means how the neurons connect to each other. And even for the macaque, this recently, a fast image and this throughput, this image technique was developed, which can actually get the wiring diagram of the macaque at this cellular or even subcellular level. However, if you look at all these experimental projects, actually it takes large amounts of manpower and time. So meanwhile, as measuring techniques developed, we know that when our brain performs functions, it can just generate different types of signals. So, for example, this two-photon calcium image actually allows to measure this over 16,000 neurons across multiple cortical areas. Of course, the measure is this the functional connectivity. And recently, also, several years ago, 2 years ago, actually, the neural pixels allows actually provide an extremely powerful and this stable measurement of this spike train, meaning when the neuron the neuron's voltage cross certain threshold, it will generate spike, and this neural pixel can measure this spike signals over the large population and also across multiple areas. And this actually is very stable and also can provide this long-term recording. So overall, this measurement for the activity takes less efforts than that of the structural connectivity we discussed previously. So our question is whether we can, infer the neural connectivity from the measurement of this neural activity, infer the structural connectivity from this brain activity measurements. So actually, this topic has been investigated by many researchers. For example, for the fMRI signal, actually, people measured this what's called this bold signal, meaning blood oxygen level dependent signal, meaning when neurons just transfer the signals, it will consume the oxygen, and then the oxygen is provided by the blood vessels. Then you can measure these indirect signals to indicate whether neurons are active or not. So for this signal, they actually compute the correlation coefficient among different brain regions, and then they compare it to the structural connectivity measured by another experimental technique called this diffusion tensor image. So the good news is actually these two connectivity. One is this correlation coefficient. We call this functional connectivity. The other is this physical connectivity, meaning neuron. Actually, they are physically connected, called structural connectivity. So the good news is these two connectivity are correlated. However, it is also found that the functional connectivity depends on different dynamic regime, meaning for different dynamics, the functional connectivity may be different. And another thing is we know our brain have different states. For example, I think you guys, are listening to my talk, so you probably pay much attention. So which means the state is different from what's you do other things. So and another thing is it also depends on the statistical methods you used. So in that sense, which means for functional connectivity, so for different dynamic regime or different brain states or different statistical method, you can get completely different functional connectivity. And however, the structural connectivity may keep the same. So in that sense, it provides the challenge for us to infer the structural connectivity from this functional connectivity. So, now our question actually is whether there exists certain statistical metric that can allow us to build a one-to-one mapping between the structural connectivity among neurons and the functional connectivity captured by such statistical metric. So the metric we want to introduce is called the Granger causality. So what is the Granger causality, or abbreviation is GC? So for example, if there's a network of n nodes, and for each node it's a time series. So what is the GC connectivity between Xi and Xj node? So first we can consider this conditional autoregression. So basically, for example, for the Xi node, which means we present the state at a t point, at the time t, using its history information and also the history information of all other nodes in the network except I and j. Then, of course, if the system is not just a linear regression, if this nonlinear, we have this prediction error, right? And also we call it a residual. And so here, A means autoregression, I means the residual for the ith node. And the reason why we have this orange part, meaning we need to consider what's called the conditional autoregression, is because we want to infer whether xj to xi, the connectivity, the GC connectivity is direct. It's not just indirect through some other nodes. So in that sense, by the regression of all other nodes, we can eliminate such indirect connection case and characterize whether xj to xi is direct connected or not. And so furthermore, we can also consider the conditional joint regression. So here, the regression is almost the same as this autoregression, except we have this cross history, meaning now we predict the xi node at the t state, also using the history information of the xj's node, which means we use more information to predict. Then we have this another prediction error or the residual. So here J means the joint regression. I comma J means the residual for the ith node by including the history of the J's information. And the GC connectivity from xj to xi is defined as a log ratio of the variance for the autoregression over the variance of the joint regression. So intuitively, you can think this way. So if including the history of the JS node, if the prediction doesn't help, which means the variance of this guy should be the same as the variance of this guy, so which means this will get this log 1, meaning 0. So which means xj to xi, there's no GC connectivity. However, if including the history of the JS node really helps to predict the state of the xi node, which means the variance of this guy should be less than this guy, then this log ratio will be greater than 0. So which indicating that there's GC connectivity from xj to xi. And similarly, for the xi to xj, we can also define this way. And so the model we consider actually is a typical model. Two actually foundation models used widely used in neuroscience. One is Hodgkin-Huxley neurons models, which that takes into account this ion channel dynamics. And the physical law is just the conservation of the current. And the leaky integral phi is a simplified version and ignore the detailed ion channel dynamics, but just set a threshold, meaning when the neuron's voltage reaches the threshold, then it shows this neuron generate a spike, and then it will be reset to some reset value and stay there for some refractory period, and then evolves again. So this just two foundation models we used. Now what's we want to investigate for simulated networks is for given this structural connectivity, meaning, for example, the adjacency matrix of AIJ, meaning the characterizing whether two neurons are connected or not. And for this HH or leaky integral phi neural model, we can run the network simulation, and then we get this voltage time series. And based on this, we can calculate the Granger causality as defined in previous discussion to get this GC matrix. Then we compare this GC matrix with the adjacency matrix. What we actually surprise us is they are highly consistent with each other. And we also tried different dynamic regime, and the consistency is always very high, approximately this 98%. So now, as we can see, the GC connectivity originally is defined for this linear regression model, but for this HH or leaky integral phi, they are nonlinear. But why we can still obtain this one-to-one mapping? So that's the question we want to address. So in order to address this question, so first let's pick two nodes. For example, this xj and xi from the network, and suppose that xj is unidirectly connected to xi with a connection strength s. And so with this physical connection, what we can imagine is once the xi generates spikes, and normally the neuron interaction occurs when the upstream neuron generates spikes, and it will cause the change of the voltage of the downstream neurons. So this actually motivates us. Maybe we can, look at this what's called a spike triggered response. So which means for the xj node, whenever it generates a spike at a certain time, we look at the voltage response of xj after it in certain window. And if xj is connected to xi, then it will just cause the increase or decrease of the xi's voltage depending on the current is positive or negative. And for another spike, we do the same thing. And then we can statistically average all this voltage response over this small window. That defines what's called the spike triggered voltage. So intuitively, as we discussed, if xj is connected to xi, and then whenever xj spikes, it will cause this some change of the xi. So when we statistically average them together, then we should see some non-trivial or non-flat pattern. So that's indeed what we see for this xi conditional on xj, and for tau is larger than 0, then we see this non-flat pattern. However, since there's no connection from xi to xj, and however we look at the spike triggered voltage for xj conditional on xi, we still see this non-trivial, this non-flat pattern. So this tells us, somehow this spike triggered response is not consistent with the underlying physical connectivity. So what's the reason? So it takes us some time that we figure out this. The reason comes from the neurons have the voltage signal has self-correlation. So for example, if we look at the two nodes, this y to x, and this unidirectionally connected. So since y is connected to x, so the history of y should be correlated to the future of x. However, since y has self-correlation, so the future of y is also correlated with the history information of y. This will actually give us this spurious causal interaction from the history of x to the future of y. So this tells us maybe we should look at certain signals that does not contain this self-correlation. So this we indeed changed our view from this voltage signal to this autoregression residuals, since the autoregression residuals and is not correlated with respect to different time. So indeed, we look at spike triggered residuals from x, this autoregression residual epsilon I condition on j. Since j is connected to xi, so we look at it. Indeed, we have this non-flat, this non-trivial pattern. However, this from I to j, there's no connection. Indeed, we see this flat, this trivial pattern. So, which means indeed the spike triggered residual is somehow is consistent with the underlying structural connectivity. So furthermore, we can just perform some asymptotic analysis to show that these spike triggered residuals actually is proportional to the time delayed correlation between this autoregression residual for xi node and autoregression residual for the xj node. And based on this, we further can show that if we discuss the GC connectivity for voltage signals, actually it is the same as that for these residual signals. So since from voltage to residual, there's a one-to-one, this linear mapping. And furthermore, we can obtain this Granger causality actually is the summation of square of this correlation coefficient, this time delayed correlation coefficient for these residuals between I and J. And finally, we can obtain this GC is proportional to the square of the coupling strength. And so then we just verify our theoretical results by taking numerical simulations. Indeed, for different dynamic regime, for example, when the neuron generates spike very irregularly to somehow the neurons generate spike almost periodically. For different dynamic regime, we always obtain this GC is proportional to the square of the coupling strength, so which demonstrates the correctness of our theoretical results. And so now we understand that the Granger causality indeed can help us to infer the structural connectivity from this neurons output signals like the voltage signals. And then the next question is whether the Granger causality can infer the structural connectivity for general nonlinear system. So here if we first look at this linear Gaussian random variables, that's just the framework of the Granger causality. We know if the xi, x2y is unidirectionally connected. When we compute the GC connectivity, it is consistent with the underlying structural connectivity. Because that's the linear regression for the Gaussian variables. That's the definition or the framework of the Granger causality. However, if we just consider this simple nonlinear transform like x squared, and we look at the x tutor and yt, the signal, the underlying connectivity from x to y is still unidirectional connectivity. However, if we compute this x tutor to y, indicates there's no connectivity, which means the computed GC connectivity is not consistent with the underlying structural connectivity. And by the way, if we consider this time-delayed mutual information, I will introduce later, and it's information theoretical metric, and this is somehow is consistent with the underlying structural connectivity. So this tells us the GC can actually obtain the inconsistent results, with the underlying structural connectivity. So in fact, for this linear Gaussian random variables, some theoretical results have shown that the Granger causality is consistent with this transfer entropy, which characterizes the reduction of this uncertainty for these random variables. And so this tells us we cannot apply Granger causality analysis everywhere, right? So this actually was proposed by a lecturer given by Granger when he received Nobel Prize in economics. He said, he gives the definition is pragmatic, meaning once you have time series, you can use it to compute the GC connectivity. And then he get many citations. Of course, many ridiculous papers appear because if you don't know the underlying the structure of the system, you cannot trust your computed Granger causality, which can correctly reflect the underlying physical connectivity or structural connectivity. Then our question is, so we know that for this time series, we can have different statistical methods to compute different causality. So our question is whether, what's the relation for different causality measures? And the second is if for a given system, so how can we choose proper this causality measure to infer its underlying structural connectivity? So, and also as a
穹苍广而横千，物和多容。星云璀璨，黑洞冥冥。
Hello? Yes. So somehow it interrupted. Okay, it's fine, right? So okay. Um so uh the next thing we want to also discuss is we want to push our theoretical results to real data. So for real data, we have to face certain challenges. For example, so previously we have discussed the Granger causality using voltage time series. And somehow in experiment, if you measure the voltage signal, it's very noisy because the signal to noise ratio is relatively low. However, if we measure this spike train, meaning once the voltage cross certain threshold, like these peaks, and this is a very has very high signal to noise ratio. So which means if we want to push our results, we should consider this spike train, meaning this digital signal is 0 or 1 instead of the continuous voltage signals. The second is in previous discussion about this Granger causality, we have to know the information of the whole network, meaning every neuron's voltage signal. However, this is impossible in real experiment since one can only measure partial information of the network, and so like this blue nodes, we call this measured neuron. Of course, there are some red nodes as these unmeasured or hidden variables. So and now we only have this measured neurons information, and we want to establish our reconstruction framework based on this partial information. So which means we should design some reconstruction technique based on this pairwise signal information. Like, for example, if we want to infer the connectivity structure between Y and X, whether it is possible we only use the information from X node and Y nodes, whether we can just infer the structural connectivity. So that's so now what we want to address, the question we want to address is the following. So first, for these pairwise the causality measures, we have different statistical methods. What's the relation between them? And the second is for this pairwise causality measures, what is the relation between this causal connectivity inferred by such statistical method and the structural connectivity among neurons. And the third is once we have established this theoretical relation and also the mapping between causal and structural connectivity, whether it is possible for us to push our theoretical results to real experimental data. And so what we discuss, this causality measure is the four widely used measures. And the Granger causality I have introduced. And also another thing is the time-delayed correlation coefficient. This I will also introduce later. And this is just standard correlation coefficient, but with the time delay. And another tool, this time-delayed mutual information and transfer entropy, that is based on information theoretical metric. And I will also introduce later. And for this signal, we are interested or we want to discuss is a spike train signal. As I said, for these signals, the signal to noise ratio is higher, and the signal is more reliable. But the information, of course, is reduced since the signal contains the component contains either 0 or 1, indicating the neuron spike generate a spike at this time or does not generate a spike at this time. And we want to infer the connectivity among neurons. Now, first, let's look at the theoretical relations between these 4 causal connectivity measures. So for time-delayed correlation coefficient, as you guys must be familiar, it's just standard correlation coefficient, but with an index m indicating the time delay, meaning the signal of y is history and the signal of x is the future. And the Granger causality also we modified the classical definition with the time delay. This the characterized by this index m. So if we choose m is 1, this will reduce to the classical definition of Granger causality. However, as we discussed in our application, so once we introduce such time delay index, so we can actually to some extent solve the issue of the curse of dimensionality when we try to apply our methods to real data. And the time-delayed mutual information, so if we say the correlation or the Granger causality actually characterizes the linear correlation between signals. So here, the time-delayed mutual information or transfer entropy with time delay. So they characterize the dependence of 2 random variables instead of the linear correlation. As we studied in the probability course, we know that 2 random variables, if they are independent, then of course they are uncorrelated. However, if they are uncorrelated, normally it does not guarantee they are independent unless, for Gaussian variables, right? So that's why for theoretical results, they show, for linear Gaussian variables, this information theoretical metric can be directly linked to this Granger causality or time correlation coefficient. However, for the general, this digital signals, the theoretical results are lacking. And if we draw a diagram for these 4 widely used causality measures, so from left to right, the method is from linear to nonlinear, and from bottom to up is whether you consider the history information or if you don't consider the history information or you consider the history information as much as possible. So we know that transfer entropy belongs to this, and it is more reliable since it is nonlinear method and also contains the history information. But the unfortunate thing is the computational cost is very high because you need to give a good estimation of this high dimensional statistical information that normally requires large amounts of data. And for the time-delayed correlation coefficient, it's linear and does not consider the history information. And the computation cost is very, very low. However, it often just gives the incorrect inference for the structural connectivity because the application range is for linear and also does not take into account history information. So the system we discussed, like the HH or Hodgkin-Huxley network, is we have this voltage signals, this evolution equation for the voltage signals. Now, since we only consider spike train, so which means we only consider when the neuron's voltage reach certain threshold, that indicates the neuron generate a spike, and which tells us actually what's the signal we consider is a binary time series. For 0, meaning the voltage is just below the threshold, and for the 1 is at this time being, the neuron's voltage reaches the threshold. And so that's just the digital signal. And for this signal, I mean, I just ignore the details. Actually, we can prove 4 theorems, and each theorem actually can link two causality measures, and with this quantitative relation and also the error estimation. And if we draw a diagram of 4 theorems, actually these 4 causality measures can be linked by this way. And we have this approximated theoretical relation. And note that for this relation between GC and transfer entropy, previous results studied it for these linear Gaussian random variables. Actually, we extended such theoretical results to these general digital signals, meaning binary time series. And so when we prove our theorems, we actually have 2 assumptions, and these assumptions are also reasonable. One assumption is the neurons, the coupling strength is relatively weak. So this is consistent with the physiological properties of the neuron, because when upstream neuron generates spike, the voltage it causes the downstream neurons change is usually small. And the second assumption is the neuron cannot generate a spike this immediately after the previous spike it generates. Meaning, if we look at the interval of these 2 adjacent spikes by one neuron, and it cannot be arbitrarily small. And this also consistent with the physiological properties, because real neurons have a refractor period, meaning once it generates a spike, it should stay this below the threshold for some time, and then it can generate the next spike. So that's we have these reasonable assumptions, and then we can prove our theorem. And once we have these theoretical relations between these different causal measures, the next question is whether we can establish one-to-one mapping between this causal and structural connectivity. And so now, since we focus on the pairwise reconstruction, and so for meaning if we pick up any 2 neurons in the network, we measured its this spike train, and then we first determine what's the time delay index. So we can compute time delayed mutual information, and we look at the curve when the TDMI reaches its peak. That's the index we choose for the time delay index, since it means these 2 random variables are are mostly this dependent with each other. And so once we have this time delay this index, then we can compute this 4 causality measures to obtain the distribution. So if we think, once the causality measures are large, then it indicates the neurons have a physical connection, have structural connectivity. And if it's small, it means it does not connect to each other. So if 2 distributions overlaps a lot, then which means the inference or the reconstruction is often bad, right? If 2 distribution are well separated, then which means the inference are very good. And indeed, for 100 Hodgkin-Huxley neural network with random connectivity, and actually we tried different connection density, they are the same, and we compute the causality measures based on the spike train. Indeed, we get 2 well separated distributions, and with this distribution in the left indicates the neurons without this direct connection, and the causality, this distribution on the right means the neurons are directly connected. And so if we don't use the information of all nodes, as we say in experiment, you can only measure partial information. So suppose we only measure 20 neurons in the network, and then we compute these causality measures, then we still get two well-separated distributions, with the left indicates the neurons does not connect to each other. The right means they are directly connected to each other. And now, so from numerical experiment or numerical results, it tells us that indeed we can infer the structure connectivity based on this causal connectivity. Now, the question is, what's the reason? What's the underlying mechanism? And to understand this, suppose we also pick up two nodes from the network. Suppose one is called Y, the other is called X. From Y to X is unidirectionally connected with coupling strength S. Then we define the what's called this delta P. Actually, it's called this spike-induced change of firing probability. Because if you look at the expression, it indicates when X generates a spike, the probability difference is one is when Y generates spikes. The other is when Y does not generate spike. So this is indeed the spike from Y induced change of the firing probability for X, right? And for this, if Y is connected to X, so this probability should also be closely related to the underlying coupling stress, right? Indeed, we know that we can prove this time-delayed correlation coefficient is proportional to or define this delta P, this spike-induced change of firing probability. And furthermore, we can show this delta P is proportional to the coupling stress, as also verified by our numerical experiments. And later on, we can prove these four different causality measures. Somehow they are also proportional to the square of this delta P. And also, since delta P is proportional to coupling stress, which means they are also proportional to the square of the coupling stress, as we discussed in previous, the results for this Granger causality. And here, for this pairwise causality matrix, and based on the spike train, we can also prove the similar results. And then you may you may feel surprised, because since the Y is connected to the X is connected to Y, they can be indirectly connected through some unmeasured nodes or hidden variables, right? So for example, for this indirect chain, suppose X is not connected to Y, X is connected to Z, Z is connected to Y. So if we don't consider the information of Z, normally it will give this incorrect connectivity from X to Y. And the second is for the confounder case, meaning they have the common driver from Z. This usually can also give incorrect connection between X and Y. But why for our pairwise reconstruction this framework, this indirect causal path does not impact or affect our reconstruction results. So this we want to show. And indeed, if we pick up two nodes, suppose they are not directly connected, they are indirectly connected through some other nodes, W. So for Y to W, there's a direct connection. We know this delta P is proportional to the coupling stress. And for another direct connection path from W to X is also delta P is proportional to the coupling stress. Then we can prove actually for delta P from Y to X is proportional to the excuse me, to the product of this delta p from y to w and also delta p from w to x, meaning the indirect, this spike induced change of the firing probability from y to x is actually the product of two directly connected spike-induced change of firing probability. And therefore, we know this delta p is proportional to the product of the coupling strengths. And so, for example, if we consider the transfer entropy, we get these results. And since we know for the transfer entropy, for the direct connection strength is often small, so which tells us this indirect causal path, since it's product of two direct ones, so that's 10 to minus 6 is much less than this direct connection strength, this 10 to minus 3. So this actually allows us to distinguish the causality between these direct interactions and the indirect interactions. And indeed, we verify our theoretical results using numerical simulations. This delta p, this indirect one, is proportional to the product of direct one. And not for this indirect chain case or this confounder case, we get the similar results. And now, for, of course, another issue you may think, when we apply our results to real data, we don't have this ground truth. So what we do is, first we get this, suppose we have these two distributions. One is distribution of connected pairs, one is indirect connected pairs. And once they are to have some overlap, so this green is true positive and the blue is true negative. The red and the yellow characterize the error part. One is false positive, one is false negative. And this straight line indicates the threshold, meaning if the causality measure is larger than this, we say the neurons have this direct connection. If the causal value is below this, we say it does not. Then we have this region of correctness and the region of incorrectness. So suppose, two distributions are very separated. We know this AUC, meaning this area under the curve. Actually, how to obtain this is you choose different threshold from negative infinity to positive infinity. So for any specific fixed threshold, you get 1 point, what is called a true positive ratio and a true negative ratio. Then you draw a dot on this curve. And since your threshold is through negative infinity to positive infinity, then which means then you finally get a curve. And the area under this is called this AUC. So if two distributions are very well separated, the AUC is very high, is 1. Of course, if the two are overlap, meaning suppose they are the same, then the AUC is relatively low. It's just 0.5. It's the same as this chance level. And then if we apply our methods, we just first get the distribution of causal values. Then we fit them by two distributions. One we say is corresponds to the neural connected distribution of causal values. One is the neuron without connection, the distribution of causal values. And for this, we can just draw different threshold, and then we get this ROC curve, and we compute the area under the curve. That is called this AUC. So if AUC is closer to 1, which means this reconstruction results are very good. If AUC is close to the 0.5, the chance level, which means the results is low. And indeed, we tried there's a neural network with different distribution from normal distribution, uniform, exponential, log normal, and the AUC is all very high. And also we try different neural models, and the results are also very good. And also we tried a different dynamic regime. Due to time, I didn't introduce it. And also this balanced state, people in experiment are very interested in. And then, so based on this theoretical result, seems our pairwise reconstruct framework works pretty good. Now we want to apply it to the real experimental data. And so the experimental data we tried is from this Allen Institute. So for the mouse primary visual cortex, they use neural pixels to measure this about this 157 units. And when the mouse look at the different visual stimulus, like a drifting grating, static grating, or natural movie or natural scene. And what we found is for different stimulus, actually the reconstruction, this AUC, are very high, are always greater than 0.95. And also for different stimulus, although the neural activity may different, and since if they can also, both of them can be used to infer the underlying structure connectivity, then we should get a consistent results between this reconstruction. So that's what we did. If we compute any two of them, I mean, under different stimulus, the consistent result, the consistency are always very high, is greater than 75%. And also we collaborate with Long Yanling's lab from ECMU. They just record the signal in awake mice, let the mice just explore the different spatial locations. And what's they interested is because the neurons can generate different these rhythms with different frequency, and they are interested in the what's called the theta rhythm, meaning they want to infer whether there are some neurons is contribute a lot to this theta rhythm, and use our method. Indeed, we can tell them indeed there exists theta neuron that contribute a lot to this theta rhythm. However, for other non-theta neurons, they don't contribute to this these causality measures. And later we also just perform this detailed realistic neural simulations and tell them that the neuron they measure is probably one specific type called CCK EC. And later they will just test our theoretical results in experiment. So, and last, I just want to also say something about our limitation of our work. So first is, since we want to infer the structure connectivity based on the spikes, so we need a sufficient number of recorded firing events, right? Because such events can, contain sufficient statistical information or statistical correlation between neurons. And also, we studied this asynchrony or this moderate synchrony. And for if two neurons are very, synchronized, meaning they always generate a spike at the same time, then there's no point you can tell which actually is cause, which is results, right? And the last thing is we consider the network that is relatively sparse. This also consistent with real neural networks. So that's a summary of our work. And so we establish theoretical results of different causal measures, and we extend our theoretical results to experimental data. And so these are also related publications. So thank you very much. Yes, that's a very good question. Actually, we know that's for real neural network. So for the relatively long time scale, our brain is the plastic, meaning the neural connectivity indeed will change. So what I mean is, even for the neuron, the connectivity strength starts to change. But suppose, let's say, the time scale of this change is relatively long, then the statistical information we need to accumulate, then it is still possible for us to infer the structural connectivity for different brain regions. And we are now also just taking this investigation on this direction. Yeah. What yes. So you can think this way. Suppose, let's say, for certain time, and we have this stable and structural connectivity, and under this duration, we accumulate sufficient information about this statistical information among neurons. Then, for this duration, we can infer the structural connectivity uh, of between among neurons. And then the neurons connect, coupling strength starts to change, right? So what we need is, so we first should detect some this what's called this changing point or tipping point when the neurons this coupling strength, because once your coupling strength starts to change, your dynamics also starts to change, right? So which means, so for this dynamics, whether it is possible for you can just detect such a tipping point or changing point. And then after that, suppose its structural connectivity reaches certain stable structure. And for that, you suppose you have this certain duration, and with and you can accumulate a large amount of this statistical information among neurons, then you can also do reconstruction on that duration to infer this new structural connectivity. So that's what I want to say. Yeah, thanks. Okay, thank you.